Thank you for the nice introduction. It has been a wonderful meeting so far, and I'm also very much looking forward to the other talks of today. So today I will talk about sensing and estimating time-varying signals. And I think it's a remarkable feature of life that many organisms can do this with very high precision, right? And even simple bacteria can measure changes in the environment with remarkable accuracy. And to illustrate this, I always like to show you this movie made by Katja Taute in the lab of my colleague Tom Shimizu here at AMOD. So here you see these E. coli cells trying to find food. And what has been inferred from experiments is that these cells can measure concentrations with extremely high precision. So it has been estimated that these cells can respond to the binding of a handful of molecules. And this I then mean in the metaphorical sense that they can respond to the binding of on the order of 10 molecules, right? And so this immediately raises then one of the overarching questions of this talk today, which is how accurately can cells actually measure chemical concentrations? Now, this question is then about measuring the current concentration, right? But mounting a response, responding to a change in the environment, taking action takes time, right? And as Thierry already beautifully explained yesterday, in this scenario, it pays off to anticipate the changes in the environment, right? And so to anticipate the changes in the environment, we have to be able to predict the future with some accuracy. Now, I think we understand that we humans can really make predictions, and Thierry illustrated that with the example of Serena Williams. Even though she's not my favorite tennis player, I think this example did drive the point home that we humans can make predictions and thereby anticipate changes in our environment. But even these little E. coli cells can actually make predictions about the future. And this is illustrated in this very cool experiment by Mitchell and co-workers, where they looked at the response to lactose and maltose. And I don't know how, but they come up with this idea that E. coli tends to see a regular temporal pattern, a temporal sequence in the order in which they see lactose and maltose. And what they argued is that E. coli typically sees lactose before maltose, right? And from this then came the idea that it might be beneficial for the E. coli cells to already express the maltose operons when it sees lactose, so before it actually sees maltose. So this is then what they tested, right? And here you see the results, right? So here you see the expression level, the promoter activity for different types of promoters. So here are the maltose operons. This is the lac gene, right? And what they found is that these wild type cells, right, that they express the maltose operon upon the induction by lactose, right? So that's what you can see here with these red symbols. When you stimulate them by lactose, they do not only express the lac operon, but they also express, in fact, these maltose operons, right? Now, of course, you could say, well, maybe there's another reason for this. Maybe it does not have to do with this temporal sequence, right? And so to test this, what they then decided to do is to evolve cells in an environment in which this particular temporal sequence is not present, right? So what they decided to do is to evolve them for hundreds of generations in the presence of lactose only. And what they then saw, and that's this panel below, is that then these cells no longer express maltose when they see lactose, right? Now, of course, the proof of the pudding is, does this, the real question is, of course, does this now also provide a fitness benefit? And so to answer that question, they performed competition experiments, and the main result is shown here, right? And so what you see here is that cells that were first grown on lactose and then on maltose, so that were first pre-exposed to lactose, that they have a growth advantage of cells that had not been previously exposed to lactose, right? And what they also found is that this is direction specific. So if you do the reverse, if you first expose cells to maltose and then to lactose, right, and then you compare the growth benefit of it to cells that have not seen this temporal sequence, then there is not a benefit, right? And what they show here is that it's actually very specific to these particular sugars, lactose and maltose, because if you do it with other sugars like galactose and succinate, you don't see this, right? So what this experiment shows is that E. coli can exploit correlations in its environment to make predictions and mount a response ahead of time. So this will then be the central question of this talk. How accurately can cells measure current concentrations and make predictions about the future? Now, before I continue, I'd like to thank the people who did the work. Julia Malaguti, Niels Becker, my former collaborator Andrew Mugler, Chris Covern, and Tom Aldridge. Now, what is now the crux of prediction, right? So the whole crux of prediction, the gist of prediction, is to exploit correlations that are in the signal, right? And this is illustrated here, right? So if you make a prediction based on the current signal value only, then you will be able to predict the future value of the signal with some accuracy, but it will not be high, right? Whereas if there are some correlations in the signal and you exploit these correlations, then you can reduce your uncertainty about the future, right? So this is really the essence of prediction. Of course, the next question then is, how are you going to exploit these correlations in the past? How can cells extract these correlations and capitalize on these correlations that are present in the signal? And to illustrate this, I've made here this little cartoon here of a cell with receptors on its surface, right? And this cell aims to estimate the concentrations of these black ligand molecules, right? These ligand molecules bind to the receptors, right? And the receptors, the ligand-bound receptors, they then constitute a noisy version of this signal, the black signal that the cell actually aims to estimate, right? And to make prediction, the signaling network then downstream has to extract the correlations that are in the signal, right? And to illustrate this idea, it's helpful to actually linearize the network, right? And then you can show that the output of this signaling network downstream, X, can in general be written in this form, right? Where S is the signal that the cell aims to estimate. C here is the noise that is added to the signal. That's the noise that comes from the receptor ligand binding, right? And this is some intrinsic noise that is present in the signaling device downstream, right? But the point here is that this signal S, this noisy signal S plus Xi, that is then integrated by the signaling network downstream, and mathematically that is contained in this integration kernel chi, right? So the idea is that the output of the network is actually an integrated version, a filtered version of the noisy input signal. Now, how accurately can cells now make predictions, right? Well, this will depend on the topology of the network, and it will depend on the statistics of the input signal, right? You have different types of signals with different types of correlations, and the optimal topology of the network will actually depend on the statistics of the signal. right? And so a few years ago already, we then asked this question: what is the optimal topology of the network given a certain statistics of the input signal? Now, of course, this question by itself is a very classical question in signal processing, right? Already in the last century, Wiener and Kolmogorov and Kalman, they developed filtering theories to precisely address this question. So what we did a few years ago is to basically use Wiener-Kolmogorov filtering theory to ask what is the optimal topology of the network given the statistics of the input signals, right? And what is very clear is that when the input signal is non-Markovian, which means that it has memory, which means that the signal in the past contains information that is not contained in the present signal. There's additional information in the past that the cell can exploit, that then it's optimal to actually use incoherent feed-forward networks and negative feedback, because these are motifs that basically allow the cell to take derivatives and thereby extrapolate the signal based on its current value and its derivative. But if, in contrast, the input signal obeys Markovian statistics, meaning it's memoryless, which means that the past signal does not contain information that is already contained, that is not already contained in the current signal, then the simple exponential filter is actually the optimal filter to extract information, right? So the accuracy of prediction, it depends on the statistics of the input signal, it depends on the topology of the signal, a topology of the network downstream that reads it out, and of course, it also depends upon the noise in the input signal, right? So what we will see today is that when the input signal is Markovian, memoryless, it still can be beneficial to exploit the past, because by averaging over the noise in the past signal, you can get a better estimate of the current signal, which then allows you to predict the future signal more accurately. So it depends upon the topology of the network, depends upon the correlations in the signal, but it also, of course, depends on how many receptors there are, how many readout proteins there are, right? Because this signal, right, is measured via these receptors, right? These receptors will then activate signaling networks downstream, right? But these signaling networks downstream are also inherently stochastic because they also consist of biochemical reactions, right? And so this raises then one of the first questions that we will address also today is, can cells simultaneously extract this information and suppress the intrinsic noise that's added to this transmitted signal downstream? If so, what resources, protein copies, receptors, readout molecules, and energy are required for this? And do these resources fundamentally limit sensing, like weak links in a chain, or can they compensate each other, leading to trade-offs between them, right? And the overarching question is, perhaps, can we derive an illuminating expression for the prediction precision, which allows us to predict the optimal design of the network, right? So, of course, this is an ambitious program, right? Because as I've argued, right, is that the accuracy of prediction depends upon the statistics of the signal, depends upon the topology of the network, it depends upon all these resources, receptor, readout molecules, time, and energy. So there are a large number of parameters, and the challenge will be to come up with an illuminating expression that gives us insight into the optimal design. Now, today we will study a Markovian signal, right? So, that's an Ornstein-Uhlenbeck process, right? Where the fluctuations decay exponentially with a correlation time tau L. And this signal fluctuates with a strength given by this variance sigma squared sub L. Now the ligand molecules will then bind to the receptor, right? So, and there will be then receptor ligand binding noise. This will be the noise added to the incoming signal that the cell aims to measure. And this receptor ligand binding noise has a receptor correlation time, tau c. Now, when this ligand is bound to a receptor, then it will drive the activation of a push-pull network shown over here. So push-pull networks are very common motifs in cell signaling. They're perhaps the most common motif in cell signaling. E. coli chemotaxis implements this motif. MAPK uses this motif. GTPA cycles basically use this motif, right? So they're all based on this idea of readout activation and readout deactivation, right? So the idea is that if a receptor is bound to a ligand, then the ligand-bound receptor will then drive the activation of the readout, right? And then this activated readout, it can actually also be deactivated by a ligand-bound receptor, right? Because of microscopic reversibility, any reaction in principle can also happen in its microscopic reverse, right? So even though a ligand-bound receptor tends to activate, let's say, tends to phosphorylate the readout, this reaction can also happen backwards, right? But once this molecule is phosphorylated, it typically then tends to also decay back to its unphosphorylated form, and then this readout has performed a full cycle during which one fuel molecule has been burned. So for example, one ATP molecule has then been turned over into ADP and the phosphate, right? And so these timescales here associated with readout activation and deactivation, they will set the effective integration time of the receptor, as I will show you in a few slides. So we will look at this common motif, this push-pull network. And what I would now like to emphasize is that this push-pull network actually constitutes an exponential filter, right? And as I already argued, these exponential filters, they're optimal for Markovian signals, the signals that we will be studying today, right? And what these exponential filters do is that they actually time average the receptor ligand binding noise. And that's illustrated over here, right? So when you linearize this network, you can show that the output of the network, let's say the number of phosphorylated molecules, readout molecules X, for example, can be written in this form, right? Where P now is the probability that the receptor is bound to a ligand, right? So this is now a stochastic quantity, right? And this is then integrated via this exponential integration kernel with this integration time tau r, right? And this again, this eta is this intrinsic noise, that's the noise added by the signaling dynamics downstream, right? But the point is really of this equation here, the point to take home is that this push-pull network allows the cell to time integrate the state of the receptor, right? And this is the whole idea of time integration, which has been known from filtering theory for a long, long time, since the 40s essentially, but which Burke and Pecil first pointed out in the context of cell signaling in the 70s, right? So with time averaging, the whole idea is that the cell estimates the concentration not from the current number of ligand-bound receptor, but rather from their average over some integration time tau r. And this is then illustrated here, right? So here you see the time trace of a single receptor switching between the ligand-bound state and the unbound state with this correlation time tau c. And then the cell estimates this average over this integration time tau r, and then estimates the concentration from the estimated average receptor occupancy by basically inverting this input-output relation, right? And if you then work out the sensing error, right, the fractional error in the estimate of the concentration, delta C over C squared, is then given by this expression. So the variance here, this variance here, that's the variance in this time average. If you assume that each receptor molecule takes an independent measurement, then you have to divide this by how many receptors you have. This dCdP, that's the inverse gain that basically determines how much an error in the estimate of the receptor occupancy, how much that propagates to the estimate of the concentration. It's the slope of this input-output relation. And if you work this out, you arrive at this expression over here, which has a very intuitive interpretation. 1 over p times 1 minus p, that's the fractional error in the estimate based on a single receptor. Tau r over tau c, that's the number of independent concentration measurements per receptor, and then RT times that, that's then the total number of independent measurements taken by all receptors collectively. So this is really the whole idea of time integration. And here I would like to point out that this mechanism of time integration is distinct from the mechanism of maximum likelihood sensing and Bayesian filtering that Thierry Morat described yesterday, right? So how do cells now do time averaging, right? How do they perform an integral, right? Now, these readout molecules, right, they are discrete objects, right? And they interact with the receptor in a stochastic fashion, right? So they take this integral by basically discretely and stochastically sampling the state of the receptor, right? And we decided to basically derive an expression for the sensing error based on this idea of stochastic sampling. And to illustrate our theory, oh, let's see, right? So here, so our theory is based on this central quantity, which is called this dynamic input-output relation, right? So here on the y-axis, you see this average occupancy over this integration time tau r, right? As a function of the current concentration, right? So here we have this ligand concentration that fluctuates over time. It has some stationary distribution of signal values, which is what is shown here, right? And this is this dynamic input-output relation, which gives the mapping between the average receptor occupancy over the past integration time and the current concentration that the cell aims to estimate, right? And the slope of this dynamic input-output relation, that's this dynamic gain, right? And it's the static gain multiplied by this factor, which basically shows, right, which shows that it depends on all the time scales in the problem. It depends not only on the dynamics of the response system, but also on the dynamics of the input signal. Right, so the idea now is that the cell uses its readout system downstream to estimate this average occupancy over this integration time tau r, and then it inverts this dynamic input-output relation to obtain an estimate of the current concentration L. So how does the cell now obtain this average, this estimate of this average occupancy? And this is this idea of sampling, right? And to illustrate this idea of sampling, right? So here again have the time trace of the receptor, of the time scale, the time trace of the ligand signal. Here you have a single receptor switching between the bound and the unbound state with this correlation time tau c, right? And here we now imagine, for the sake of the argument, that we have these unphosphorylated inactive readout molecules that now then will interact with the receptor, and each time a readout molecule interacts with a ligand-bound receptor, it will then be phosphorylated. Whereas if a molecule interacts with an unbound receptor, it will not. Right? And what you can now see is that the fraction of active readout molecules, right, shown here, right, that provides now an estimate of the average occupancy over the past integration time. Now, of course, in order to work out what the error is in this estimate, right, we do not only have to consider the process of taking samples, which is illustrated here, right? So here is illustrated the process of taking samples, right? But we also have to recognize that these samples can decay, right? These phosphorylated molecules, they can also be dephosphorylated, which will lead to the erasure of a sample. Moreover, as I already emphasized before, right, it can happen that a phosphorylated readout molecule will interact with a receptor that's actually bound to a ligand and then be dephosphorylated, right? So while normally a ligand-bound receptor is mapped onto a phosphorylated readout molecule, it would then be mapped onto an unphosphorylated readout molecule, right? And that would screw up the coding, right? So to protect the coding, this signaling network has to be driven out of equilibrium. Last but not least, we also have to take into account the dynamics of the signal, right? And that's shown here, right? So here you see two trajectories that both have the same current signal, right? And this is the signal that the cell aims to estimate, right? We imagine here that the cell wants to estimate the current signal. For Markovian signals, this is actually also equivalent to predicting future signals. Right, so okay, so the point here is that a given current concentration, which the cell aims to estimate, there's actually a whole ensemble, a whole zoo of trajectories in the past, right? And each of these trajectories in the past will give rise to a different average occupancy, right? And that will thus lead to a systematic error in the estimate of the current concentration, right? And so if we now bring this together, right, then we can derive this expression now for the relative error, right? So that's the error delta L, the absolute error in the estimate of the concentration divided by the width of this distribution, right? And so this is basically a measure for how many distinct signal values the system can actually resolve, right? Or the inverse of that, I should say, right? And this relative error, as before, is given by this variance in the estimate of this average occupancy, which is this quantity, divided by the slope squared, which is this dynamic gain, and then we still have this variance from this definition. And if you work it all out, then you arrive at this expression for the relative error. And what you can see is that it consists of two terms, the sampling error and the dynamical error. The sampling error I will discuss in more detail on the next slide, right? Here I will focus on this second term, the dynamical error. And what you can see here is that this term only depends upon timescales, right? So it does not depend on how many receptors there are, how many readout molecules there are. It also does not depend on how much energy is devoted to signaling. It only depends on the timescales in the problem. And it really comes from the fact that a given signal, a given current value actually corresponds to an ensemble of trajectories in the past, which will give rise to a systematic error that the cell cannot eliminate by taking more samples. This dynamical error can only be reduced to zero by reducing this integration time to zero, right? Because if you reduce the integration time to zero, then basically the estimate of the past corresponds to the estimate of the current signal, right? So the dynamics shows up here via this dynamical error, but it also shows up here in this prefactor of this sampling error, because indeed this term, or these two terms here in green, right, they describe the error in the estimate of the occupancy due to the sampling, but how that then propagates to the error in the estimate of the concentration, that of course still depends on the slope of this relation, which is this prefactor, right? Okay, so what is now these two terms here in green, right? And here you see this quantity Ni, right? And that is the number of independent measurements, right? So that's the number of independent samples taken over the receptor over this integration time, tau r. And it's given by this expression over here, right? And this has a very beautiful and simple interpretation, because what is n dot? N dot is the rate at which the readout molecules interact with ligand-bound receptors. P is the probability that a receptor is bound to a ligand. So n dot over P is the probability that, so n dot over P is the rate at which readout molecules sample the receptor, be they ligand bound or not. It's the sampling rate. Tau R is this integration time. So this product here is just the average number of samples over this integration. Now, as I mentioned, not all samples will always be reliable, right? If the network is not driven out of equilibrium sufficiently, this reaction can happen in the wrong direction, corrupting the coding. And this effect is contained in this quality factor. It's indeed 0 when the network is not driven out of equilibrium at all, and it goes to unity when it's strongly driven out of equilibrium. And then the last factor here describes how independent the samples are, right? And it's only when the readout interacts with the receptor over a time scale that is longer than this correlation time that only then these samples become independent, and then indeed this fraction nicely goes to unity, right? So even though this is a relatively long expression, right, it really reveals the resource requirements necessary for sensing in terms of collective variables that have a very intuitive interpretation. Now, in the remaining minutes, I think I still have about 7 minutes. I would just like to highlight a few results. So the first result is highlighted here, right? And that is that the receptors, readout molecules, and power are all fundamental. They're all necessary for sensing, and they cannot compensate each other, right? So what you see here is the mutual information that's basically related to the relative error, right? So it's high when the relative error is small as a function of the number of readout molecules for different numbers of receptors, right? And what you can see here is that in this regime, this mutual information, this sensing precision is independent of the number of receptors. In this regime, it's completely limited by how many readout molecules the system has. In this regime, the system can only increase the sensing precision by increasing the number of readout molecules. But if you then increase the number of readout molecules further, then at some point the readout molecules are no longer limiting, but the receptors become limiting, right? And so only in this regime, the sensing precision can only be increased by increasing the number of receptors, right? So clearly, readout molecules and receptors are both important. And here, the same is shown for the precision as a function of the power and the number of receptors. So when the power is low, right, the precision is independent of how many readout molecules you have, how many receptors you have. In this regime, the precision can only be increased by increasing the power. Whereas in this regime, where the power is really abundant, here only the precision can be increased by increasing the number of receptors. Now, what we then could also show is that this optimal integration time actually depends on the number of receptors, right? And that makes intuitive sense, right? Because the number of receptors determines the sampling error. The more receptors you have, the more instantaneous measurement the cell can take, the less there is a need for time integration, and the more beneficial it will then become to reduce the integration time, because then you can increase the gain and reduce the dynamical error. Then what we could also show, what also follows from this, is that actually in an optimal sensing system, the number of independent concentration measurements at the level of the receptor, which is given by the number of receptors times the number of independent measurements per receptor, tau over tau c, that also equals the number of readout molecules to store these measurements, which also equals the energy in units of kT to store them reliably, right? And this here, right, when the system obeys this relation, it precisely sits at this sweet spot, right? So if you would be here, right, if the cell would be here, then clearly it does not have enough readout molecules. The sensing precision is really limited by the number of readout molecules, whereas here it's really limited by how much the receptors there are. And this principle, this rule here, precisely corresponds to this intersection. And this is actually then, this then actually gives a very concrete prediction, which we which was actually which we could actually confirm by just looking at the literature, because cells express these readout molecules. E. coli expresses key Y and these key A dimers, which are a proxy for the active receptors, depending on the growth rate. And what we would predict is that there's this linear relation with a slope set by this integration time over the correlation time, which also has been measured independently, and this really fits beautifully. So then the last slide that I would like to show, and that will probably take me 2 minutes, is that we also use this now to ask this question: how accurately can these E. coli cells now measure these time-varying concentrations, right? So E. coli performs these runs and tumbles, right? So it sees a fluctuating signal with a correlation time that is actually set by the run time. And if the cell then swims in an exponential gradient, then the strength of these fluctuations, the variance of the input signal, is then basically set by the length scale of this gradient, right? And our theory then predicts that this optimal integration time depends on the number of receptors, the strength of these fluctuations, and the time scale of these fluctuations set by this runtime, right? And this figure then shows the results. So here you see the optimal integration time as a function then of the steepness of this gradient, right? So as emphasized here, right, the steepness of the gradient, that basically sets the strength, the variance of the input distribution, right? And what you can see is that when the gradient becomes more shallow, the optimal integration time increases, and that makes perfect sense, because when the gradient is more shallow, time integration becomes more important to estimate this accurately. Moreover, and more importantly, when the gradient is shallow, then the variance, the width of the distribution becomes smaller, right? And then it does indeed become more important to measure accurately. And so then this integration time increases, right? And what's interesting is that the measured integration time is over here, which then suggests that indeed E. coli has evolved to sense these very shallow gradients. And then the last question is, can cells now also measure this sufficiently accurately, right? And so to that end, we then actually measure the signal to noise ratio, right, as a function of the steepness of the gradient, right? And indeed, steeper gradients lead to a larger signal to noise ratio, right? But we would argue that in order to measure the signal accurately, the signal to ratio should be on the order of unity, right? And as our analysis predicts, with that, it can actually still measure these very shallow gradients, right? So what we would like to argue is that this signaling network has evolved to optimally sense shallow gradients. Okay, with that, I wrap up. So non-equilibrium sensing requires receptors and their integration time, downstream molecules and energy. These resources cannot compensate each other. All of them are fundamental, right? This is in contrast to static signals. I didn't really dwell on that, but you can imagine, right, that for static signals, time can be traded against power and can be traded against receptor. But for these time varying signals, time and power separately become fundamental. The optimal integration time depends on the strength of the fluctuations, their correlation time, the number of receptors, and the correlation time of the receptors. And what I tried to argue also is that E. coli obeys the design principles as predicted by our theory. And with that, I would like to thank you for your attention.
Yeah, thank you for this nice talk. So we are slightly delayed, but if there is a quick question, we can take it. Yes, so Ayan?
Yeah, Professor Tenolde, it's a very quick question. So you said that for the accurate prediction, the noise statistics and the topology of the network are two important things. Now, we do know that cells do kinetically proofread the incoming signals. And for that to happen, it has to be reversible in nature. But if you have an incoherent FFL, it has a branched pathways. But reversible signaling has to be in a very, Uri alone has showed that it has to be like one step cascade type of thing, so that it can be reversible in nature. So
yeah, and the question is?
The question is, so is it not something important to include in the topology of the network, or maybe the network structure is something related to, as you showed previously in your paper, the proper multiplexing and demultiplexing of the signals?
Well, so I think multiplexing is really an interesting question when you have many different types of signals and you want to discriminate between these different types of signals, right? Then also kinetic proofreading will become extremely useful. But that's not a question that we have addressed here, right? So here we look at one type of signal, and if you have memory in the signal, if it's non-Markovian signal, so let's say an underdamped particle in a harmonic well, for example, that has some memory, some persistence, right? Then you really would like to take derivatives to do extrapolation, and then incoherent feed forward, negative feedback is very useful. But if the signal is just a Markovian signal, then a push-pull network has actually an optimal topology. Yeah, so I don't think that then kinetic proofreading would be really necessary.
Okay, thank you. So I will ask the other people who have questions to